morning, everyone. Welcome to Clark Lindgren's second class on synapses and self. We're happy to have you here this morning. I'm Joanne Bungie, and my job is to remind you to silence your cell phone and to turn on your tea coil. We appreciate you coming out. We didn't realize it was going to be icy this morning. We felt we had really dodged the ice storm almost completely, but it did prompt me to remind you of our procedure for when we have to delay a class because of the weather. If we have weathers on Tuesday afternoon or Wednesday morning so that we can't have class on Wednesday morning, then we will send each of you an email if we have your email address. Now, if you don't have a computer and don't have an email, perhaps you could work up a system with a friend who does have email to let you know when they have received an email from us. It'll also be announced on KGRN, and it will be on our website as well. So that's for any time when you have a question about whether or not class is going to be held because of the weather. That's how you can check. Also this morning, we want to remind you that the next 2 weeks we have a time change for our class because of Clark's teaching schedule at the college. Next week we will begin at 9:30 in the morning, so come about a little after 9. Coffee will be here, so you don't have to worry about that. And then the class will conclude at 10:45. And we ask you to please not have any questions for Clark after class because he needs to make a speedy getaway to get back to his day job. So we are happy this morning to have Clark's second class on synapses and self. Professor Clark Lindgren, who is Professor of Biology at Grinnell College and Iowa's Professor of the Year. Glad to have you, Clark.
Thank you. Can everyone hear me back there? Good. All right. So you knew what was going to happen. Quiz. So here's the synaptic vesicles with, I'll just do this. Actually, let me, hold on. All right, so we have the synaptic vesicles that are filled up with neurotransmitters. We have, I don't know, I'm filling them all up. And then we have receptors over here that will act like a lock and will interact with the neurotransmitters like a key interacting with a lock, opening it up. How do we get this thing started? So right now it's just sitting there. How does the synapse, what initiates the events that happen at a synapse? So calcium, but before that, there's, right, good. Okay, so there's some electrical event that starts the whole process off, and we call that an action potential, which propagates like dominoes falling, really the best analogy, and all the way down to the end of the axon. Now, there are 4 ways that can start. So what are the 4 mechanisms that can get a synapse started, or actually, they can get those electrical events started? Well, the first is a neurobiologist can do it if the neurobiologist has the proper tools. So, like in my lab, that's basically, when I study the synapse, I'm the one who basically decides that it's time for the synapse to synapse, and do that by electrically stimulating it. But in nature, there's really 3 ways. So the neurons right now in your head, and you have roughly 80 billion of those, most of them are probably firing action potentials right now. You're alert, you're paying attention, you're getting action potentials. There are basically 3 reasons that those cells are firing right now. The most common reason that any one of your neurons is firing is because it was activated by a synapse onto it. So it's firing because it was activated at another synapse. Okay, that makes sense. So most of your brain activity is triggered by the activity of other most of your neurons' activity is triggered by the activity of other neurons which are synapsing onto it and causing it to fire. So that's really the major reason. The second reason that a neuron can be firing is because it's a sensory neuron. And in that case, the neuron is firing because it's transduced some stimulus in the external environment, like touch on the skin, or light, if we're talking about the eye, and it converts that energy into an electrical signal. Okay, so that's the second reason. And then the third reason that a neuron can be active, and this is definitely the very few neurons are in this category, but there are some neurons that are automatically active. They activate on a regular basis. They're called pacemaker neurons. And the reason you're breathing right now is because you have those neurons in your brainstem. And they're periodically firing, and then roughly once every few seconds they fire. That's actually what allows you to breathe. So just so you understand the context of this, we're particularly looking at what happens after those events are initiated in one way or the other, and how the information then moves across the synapse. But those are really the ways that it comes about as far as we know. All right, so then what's next? So the action potential, this falling domino, reaches the end, and then somebody said it. What's the next thing that happens? Calcium. Calcium, right. So there are special proteins that are sitting right here that are proteins that allow calcium ions to move. And so those proteins get activated, their gates open, and we get calcium moving in. And then what happens? What does the calcium moving in then do? Somebody's got to be bold and shout it out.
Release to the gates.
Okay, so the calcium signal causes the synaptic vesicles, which contain the neurotransmitter, to actually move up, fuse with the membrane, and then they release the neurotransmitter out into the synaptic gap. Okay. Then what happens? Which gates? So somebody says the gates open. Okay, so the neurotransmitter binds to these proteins over here, which open their gates, and let's pick a different color. Okay, and that lets ions move in over here, and that produces what's called a synaptic potential, which means a voltage chain, or potential, electrical potential that's generated at the synapse. So this is called the synaptic potential, and it's the electrical change which will then, under the right circumstances, cause this postsynaptic cell now to fire its action potential, which then propagates down to the next synapse. Okay. What have I missed? What do we need here to make this?
But the action potential goes on the cell membrane again, right?
Yeah, I wasn't being probably very precise there. So you're right. So what would happen is the synaptic potential, if you really want to get into the details of how this works, but sure, but the synaptic potential will actually trigger these voltage-gated channels here. So you're right. So actually, the action potential would move that way. That's kind of what you wanted me to do. Yeah. And you were exactly right. I will erase this. Right, and then it'll continue on, and it will go to the next place. What's missing here?
Are you talking about the reuptake?
So there's reuptake, but this is the neuromuscular junction too. So before the reuptake, there's the
Electronic?
Remember? You don't remember the name?
I don't remember the name, but it's the things in the middle that push them back.
That's good enough. Yeah. That stuff right there, right? So what she said is that stuff there that pushes it back, or something like that. But what it is it's an enzyme. It's called acetylcholinesterase. So you're not going to remember that, but ACHE is probably good enough. Okay, so what it is the neurotransmitter is called acetylcholine. And so we abbreviate it with an A that stands for the acetyl and then the CH for choline. Okay? And that, the two parts of that molecule, the acetate and the choline, are held together by a chemical bond, and this enzyme just clips it, breaks it apart. So it splits apart the acetate and the choline so that it can no longer work. It's like taking your key and just breaking it in half, and now it can no longer work on the door. Then the choline then does get taken back up again and recycled, but that actually happens later. So the turning off part happens by chemically breaking down the acetylcholine into its two components. And I think I mentioned last time that both because of the efficiency of this enzyme and where it's located, which is right between, the neurotransmitter, the acetylcholine molecules have to make it through before they can even bind to the receptor the first time, and you can estimate that about half of them are degraded before they even make it to the receptor the first time. What that means is that these molecules will come over, the 50% that make it through, they'll bind to these receptors over here, and then they'll come off because they basically don't bind tightly. It's probably a key is entirely the best analogy there, because it's a key that comes in, but then it kind of falls back out again. So you have to kind of keep putting it in if you want it to keep opening the door. So the key falls out, and then when it falls out, it gets broken down. It has a chance to get broken down. So what it ends up meaning is that the neurotransmitter, the acetylcholine, gets broken down really quickly after it's released, because there's just this enzyme here that is very efficient. It's located in an optimal place for breaking down the acetylcholine. And remember, we said it turns off the synapse, and that's an important feature that synapses have to have. It's to work as a signal, you have to turn on, and then you also have to turn off. All right, I think that's it. So, oops. We're not there yet. Yeah, I'll go the slow way. Shoot. Let me see. Should have been set up better. All right, so where are we? We are here. Okay, so how does these experiments that were originally done to work all of this out were done on the frog neuromuscular junction, which isn't terribly different from the neuromuscular junctions that are found in all animals, including humans. So how does the human neuromuscular junction work? And basically it works pretty much the same way. And I wanted to emphasize this point because I don't want to overstate the similarities between different organisms, and that was maybe, I might have gone a little bit overboard last week in emphasizing to you that squid and frogs and humans are more alike than they are different. And I highlighted that because that probably was a concept that might have been a little bit surprising or a little new to you. But sure, they're not identical, right? We're not identical to frogs, so we have differences. And so, although there's a lot of similarities, there also are differences. And so, especially when a field initially starts off and we're trying to learn we're trying to just get like the outline and the general gist of how things work, these similarities are perfectly fine. When you start getting into the details, then you find, oh man, this worked in mice and it doesn't seem to work in rats. Like, go figure. Like, they seem like, to us, they look kind of like the big version of the other one, but in fact, they're different, and so things that might have worked in one don't work in the other. And so when you're looking at medical applications of some of these things, you obviously have to test it in humans. You can't just sort of assume that if it works, it's magic in a mouse, it's going to work in a human. But that certainly is going to give you a pretty good clue. So anyway, I just wanted to make sure I reset that a little bit. So we're not identical to squid, but we're actually more similar than we're different. So the human neuromuscular junction works mostly the same. Few differences. There's a slight difference in the anatomy, with the structure of what the synapse looks like. And there's some differences in terms of the different molecules that are involved in regulating what's going on. But the basic fundamental things we just went through are identical between frogs and humans. So what can go wrong? What could possibly, now that you have this background in synapses, especially synapses at the neuromuscular junction, what can go wrong? So let's talk about the three things we talked about, George.
Well, again, I can't remember the name of that inhibitor there, but if it keeps firing, you get contract and your muscles don't work.
Okay, so one possibility is the inhibitor might get messed up in some way, and so the synapse won't turn off. Right?
Yeah.
Okay. What's another possibility?
Why not turn on?
And why would it, what would be the reasons it might not turn on?
Calcium deficiency?
Well, so, sure, so one possibility would be that there's a calcium deficiency. That probably doesn't happen ever physiologically, because to get to the point where you would start noticing effects at the synapse, you'd be dead well before that, because you'd have to get the level so low that it would cause other problems. But you're on the right track. So you could have something that interferes with the amount of calcium in your bloodstream. But what else? It's related to that. What could also be messed up, or could become messed up, that's related to calcium?
Too much HCA, HE? Much of that.
That's a different topic. Let's stick with calcium. So in order for calcium to do what it needs to do, it needs to have, there has to be these little red blobs here. So those little proteins. So these proteins, and so diseases, most of the disease processes that exist probably involve a defect in some protein or proteins. Proteins are the things that, I mean, we think of proteins as the stuff you eat, right? And you just sort of lump all proteins together and think, they're pretty much all doing the same thing in terms of our diet. But the way that cells get put together, the way your body gets put together, is because you have very, very specific proteins that do all the different things that you need them to do. So the level of the cell, that's really, really critical. So if you have a very, very specific protein that only lets calcium in and has its gate that opens when the cell is excited, things can go wrong with it. And so that's another place where you could have a problem. So you should see how we're doing this. What's one other possibility? And I'll point to it. Oops, I'll try to point to it. What about right here? There's another protein. So that's a different protein that's acting as the lock on the postsynaptic side. So that can get disrupted in some way and can cause problems. Okay, it's not really terribly complicated. So how does that relate to things we know about in terms of human fallibility? All right. So one condition, has anyone heard of this? It's called myasthenia gravis. Do you know anybody with it? Okay. So it really is it means grave muscle weakness. And so people with myasthenia gravis will start off, maybe one of the most common symptoms is they'll actually have blurry vision. It's because the neuromuscular junctions that control the movement of their eye in the orbit actually are the junctions that first become susceptible, and so they might have blurriness or whatever, but it eventually progresses to much more major muscle weakness. It can actually be serious or grave. Where do you suppose causes that? Take some guesses, but I won't make you guess too long. So that, people with myasthenia gravis, they have lost those receptors that I pointed to on the postsynaptic side. How do they lose them? It's an autoimmune disorder, so they actually, their own immune system, for some reason, stopped recognizing those as being their own, and so treated it like a foreign pathogen, some foreign agent, and started attacking it, and so they actually started losing their acetylcholine receptors. Well, if there's fewer of those acetylcholine receptors, it makes sense that everything being equal, you're going to have less of a muscle response. It's going to not be as great. Okay. Lambert-Eaton syndrome, anybody heard of that? Much less common, I wouldn't have expected so. Named after two Mayo Clinic doctors who described it. In this case, it's also muscle weakness, but now it comes about because the calcium channels have been destroyed. And they get destroyed in the same way. It's an autoimmune disorder, and people will, again, for reasons that often are never completely understood, they will start to target specific proteins that they would normally do. So in that case, again, you get muscle weakness, but it's because now those calcium channels have been disabled, and so now, everything being equal, you don't get as much calcium entering the nerve terminal, you don't get as many vesicles releasing their neurotransmitter contents, and so you get another example of muscle weakness. Okay. And then the third one I was going to mention here, and this is not really a disease, but George mentioned this acetylcholinesterase, and so that's how nerve gases work. That's how sarin gas is fatal. it blocks the acetylcholinesterase. It blocks the enzyme that breaks down acetylcholine. And so what's going to happen to a person who's been exposed to sarin gas and their acetylcholinesterase is inhibited? What's going to happen at their neuromuscular junctions? They're just not going to turn off, right? They're going to turn on, they're going to stay active. And what physiological process requires the regular relaxation of muscles? Breathing. So breathing, to breathe in, you contract your diaphragm, pushes down expands your rib cage, that allows your lungs to fill. But then to exhale, you have to relax that muscle. It comes back up to its original position. So if it remains contracted, it can't continue to open and close, and so you basically suffocate. So sarin gas is basically kills people if they get enough of it, and paralyzes enough of their respiratory muscles. It causes them to die. Now, there also are, side effects of sarin gas if you're not right in the immediate vicinity and you don't get a high enough exposure to actually cause that effect. Then there's other places where you can have problems because for the same reason. But usually the real danger of sarin gas is to basically asphyxiate people. Okay? Sorry. Pardon?
Would it be possible for an outside influence to disrupt the electrical system of the body? Like microwaves?
So the question is it possible for outside influence, an outside signal to influence the electrical events? Because that's one of the things we didn't highlight. And the answer is yes. It is possible to do that. It takes a really pretty large, like a very large magnetic or electric field to do that. And so like being concerned about microwaves or cell phones, that really hasn't panned out, and probably the reason is those just really aren't very powerful. But a really interesting technique is called transcranial magnetic stimulation. I forget who was talking to me earlier about that. Was mentioning that. It basically, you take this electromagnet, and you place it on the person's head and turn it on, and it can actually either activate or inactivate neurons, and it's working by altering the electrical activity. And so if you go to YouTube, there's these great videos where people, they're just sitting there, normal, college student, this is being tested on, and once they turn it on, they'll start moving their arms involuntarily, or they will actually shut down activity as a result of that. So the answer is yes, but it has, and in that case, it's focused really very specifically on a small part of the brain. So
Could make a weapon.
a weapon could be a weapon, maybe, but it would have to be really, really strong. So we don't have anything that's quite that strong.
Paralytics like curare and succinylcholine, is that their mechanism?
Good. So the question is paralytics like curare, succinylcholine. What they do is they block the binding of acetylcholine to the receptor. So and so basically has the same effect as myasthenia gravis, where you basically are, in that case, you're physically removing the receptors. In cases of curare, you're basically blocking it. Curare, have you heard of that term before? So it's was used by various indigenous peoples, like in South America. They would tip their arrowheads with this material, and then when they would shoot their prey, the prey would become paralyzed, and so they wouldn't run as far, so they would actually be paralyzed, and so they would more or less drop where they were shot, so they didn't have to actually chase them down and wait for them to bleed to death, which would be normally what you do. It's used both. I use curare on a sort of regular basis to paralyze the muscles that I'm working on because I don't want them to contract. It's also used, if you've ever had general surgery, and unfortunately many of you probably have, you've gotten a drug like that. Actually, pancuronium is the one that's usually used. Because you need to be paralyzed when you go under surgery, even though you're anesthetized, even though you're completely unconscious. If the surgeon started to cut into you, even being completely unconscious, you'd probably jump off the table or at least have some sort of very extreme physical reaction to that because of the reflexes that are just built in. So that's why you have to have a respirator to breathe for you, is partly because your muscles are paralyzed when you go for general surgery. But that's for the surgeon's benefit, not for yours. That's just the surgeon doesn't have a moving target to try to. Okay. What else could go wrong? I knew we wouldn't get this one, so I basically put this down. So what's another place, let's go back to our model. Maybe I just didn't leave enough time, but I guess right. So what's one other place where we could have a fault, where the process could go awry? So we talked about the calcium channels. We talked about, let's see, does this work? Yeah. So the calcium channels, we talked about the neurotransmitter receptors, we talked about the acetylcholinesterase. Can you think of any other step that we missed? We've talked about the interfering with the electrical events.
Synaptic?
Yeah, so the synapses. So this process of releasing the neurotransmitter requires several other proteins that are specific proteins that act like little machines. And they grab the vesicles and they move them over and then they allow them to fuse with the membrane. And so that's one other place where the process can get messed up. And we call that like the release machinery. So there's all sorts of proteins around here that can also be defective.
Is the protein that we eat connected with this protein that you're talking about here?
Yes, sort of. So the protein, so the question is the protein that we eat connected to the proteins that are here? Yes, but what happens is the proteins that we eat, we break them down to the building blocks, which are called amino acids. So amino acids, and there are roughly, well, there's 20 of them and then some modifications, but they get put together in these long chains, and that's actually what a protein is. It's just a bead. Think about beads on a necklace. Each of the beads is a different amino acid, and those actually make up the different proteins. So if you eat cow meat, you're getting those proteins that were in the cow or in the asparagus, if you want to be vegetarian about it. In either case, you're getting those same proteins, but first you break them down to their building blocks, and then your cells remake them up in the way that's specific for their recipe that basically makes them a human protein for the specific place you are. Does that make sense? So you can't eat meat and then take the protein that's in the muscle and then immediately put it into your protein and have it work. It has to be completely broken down and then built back up again. So you've heard of botulism, caused by the bacterium Clostridium botulinum, and what happens? So botulism, of course, if you don't properly stew your tomatoes and kill the bacteria before you can them, and then you let them sit in this anaerobic environment for a long time, and then decide to open it up and eat it someday, you can have this rather surprising shock of getting botulism, which is actually, it really can be fatal, actually. There was an Iowa State student a few years back, I don't know if you remember that, who decided he'd basically lunch on his stir fry that he had, which is a perfectly reasonable idea, except this stir fry had been sitting in his cupboard for several months before he decided to eat it. So he got botulism and became completely paralyzed. He had to be put on emergency breathing. He had basically be in intensive care. It took him several months to recover, and he ended up having to undergo physical therapy eventually just to learn how to walk and move again. So it completely shuts you down. How does it work? Well, the botulinum toxins, and there are several of them, so they're all labeled right here. So this is the synaptic vesicle filled with acetylcholine. This is the presynaptic membrane where the vesicle is going to fuse. And these are 3 of the proteins that are critical for that process to occur. And each one of these arrows points to a place where one of the different toxins within botulinum, so botulinum has like 8 or 9 specific toxins that all work at different places. And so they cleave these proteins and then they prevent the vesicles from releasing their neurotransmitters. So it's a pretty severe way to mess up your synapse, is to go in and prevent the vesicles from releasing their contents. Okay, so that's how botulism works. I'm fascinated by these toxins that are produced by bacteria or by other organisms because I've never known a case where the toxin that they release only targets one thing. And that one thing would be fatal. So for example, if Clostridium botulinum only produced, botulinum toxin A, it'd be fatal. Okay, if it clipped SNAP25, the whole thing would fall apart. It wouldn't work. But there's this arms race that goes on over the years, and so these bacteria or these other fungi or invertebrates that produce these toxins, they just start producing more and more and more and more of them. And really, literally, the word overkill is a really good example, as a good term here, because they can just completely mess up all of the proteins that are involved, and they tend to stay messed up for the time it takes for the cells then to clear all that out and sort of restart remaking those proteins. So those are the that's another example of where things can go wrong. Botox, yes.
What about stuff like, the classic stepping on the rusty nail, getting, what is it, tetanus toxoid or rabies? Do either of those act in that way?
The rusty nail, which causes...
With tetanus, you get your tetanus shot, and there's the tetanus toxoid, and people get stiff man syndrome.
Right. And I think, I'm not 100% sure, so I probably shouldn't speculate, but I think it doesn't work the same way botulism works. It actually does the opposite. It actually sort of activates synapses and doesn't turn them off. Is there a reason I'm not? I'll have to reset this, I guess.
You wiggled it and came back up, so.
I wriggled it? Okay. There we go. So how many here have had Botox? No, you don't have to. So what is Botox? It's used cosmetically, and it actually gets rid of wrinkles. Now, how in the world this person here is allowing somebody to inject needles into various places in her face? How does that get rid of wrinkles? Pardon? Sure. So it's basically, Botox is made up of one of those toxins. I can't find this. It's made up of one of those toxins, and it paralyzes the muscles, and it works locally. So basically, it paralyzes the muscles that would normally constrict up and cause wrinkles. Okay. So that's how Botox works. Botox is also has other medical uses, and it actually was originally discovered as a cure for strabismus or cross-eye, because kind of for the same reason it would relax the muscles that were actually causing the eyes to not properly separate. And then later on, it became Allergan is the company that actually makes this, and it I'm gonna have to just wing it from here on out. And what it does is Allergan is a pharmaceutical agency that makes eye care products, and so they actually that's why they actually were the ones who sort of got a hold of this, because it was being first used by ophthalmologists to treat people with cross-eye. Then, of course, it exploded with the. This is going to be. All right, let's try this one more time. Then the use of Botox just really exploded with the cosmetic use of it. So the company went from being a rather small company to actually having sales on the order of $1 billion a year with the explosion of the cosmetic use. And then more recently, as in the last couple of years, there have been off-label uses. I don't know if you guys get time, but this was in last week's issue of Time, talking about the explosion, the second explosion of use of Botox for various off-label uses, and treating things like depression, heart trouble, migraines, erectile dysfunction, back pain, sweaty palms, drooling. I guess we only get like 10 seconds at a time, so I'll talk fast. But you may know this, that once the FDA approves a drug, then a physician can prescribe it for things that it wasn't approved for. So as long as the FDA approves a drug for, let's say, strabismus, the physician can basically decide to use it off-label, if that's not illegal at all. George?
This is totally on Botox, but since you're having problems with going on and on, I'll disrupt you again. How about a really heavy blow to a muscle or a muscle structure? Will that affect the synapses?
So the question is, will a really heavy blow to a muscle affect the synapses? Not that I know, not more than you might, I mean, not in any way that other than if you happen to have the blow that was specifically on the muscle where the synapse was, I suppose you could cause some damage there. But there isn't a specific thing related to that, as far as I know. So anyway, Time Magazine reported on this because it's, I don't know if I would describe it as a problem, but it's actually caused Allergan's sales to jump from $1 billion to $2.45 billion over just a couple of years, and most of the uses now are not for cosmetics, but for these various off-label uses, which, as the article points out, you know, sort of both good and bad. I mean, some of the uses of Botox in these off-label ways are effective, but they're kind of physicians going rogue, and they're sort of going off what all the research has been used to support. In some cases that's helping, but in other cases, you can sort of imagine that's probably just confusing. But anyway, that's all Botox, and it's working by knocking out this process of synaptic transmission. All right, a couple more things about synapses. 5 minutes? Okay, so first of all, they're not all excitatory. And so the synapse that was first discovered, the frog neuromuscular junction, is an excitatory synapse. You excite your muscles. You don't activate them by inhibiting them. They inhibit on their own. So it's all excitatory. In the rest of the body and throughout the brain, there are actually more synapses that are inhibitory than excitatory. You actually have more inhibitory synapses in your brain than you have excitatory. They work the same way, the process is identical, but they inhibit as opposed to excite. And what happens is that inhibitory and excitatory synapses play sort of this tug-of-war game on the postsynaptic targets that they're synapsing on. And just to play dangerously here, okay, I'll go back to this. Where did I put that? Okay, here it is. Okay, so this is the situation that you have that's more typical of a synapse that you have throughout your body. You have a postsynaptic target, a postsynaptic cell, which is here. So this is the postsynaptic. But it's receiving synaptic inputs. Remember, on average, about 1,000 to 10,000 synaptic inputs. So it's getting lots and lots of synaptic inputs. Some of them are excitatory. So, and the most common excitatory neurotransmitter other than acetylcholine is a neurotransmitter called glutamate. I'll just mention that here. Have you heard of the term glutamate? So it's one of those 20 amino acids that builds up proteins. It's also used as a neurotransmitter. In fact, it's the most common neurotransmitter. Monosodium glutamate, which is sometimes like Chinese food will have an overabundance of. The reason it causes problems is because the monosodium glutamate that you get activates your glutamate receptors throughout your body and mimics what the normal neurotransmitter glutamate would be doing, and so it can produce some toxic effects because of that. So there's glutamate, and it pretty much works the same way the acetylcholine does. Then there's also inhibitory synapses, and the most common inhibitory neurotransmitter is one called GABA. Stands for gamma-aminobutyric acid. And what it does, it gets released exactly the same way, so all of the same processes that we talked about, the calcium channels and so forth, cause it to be released. But the difference is that the GABA receptors inhibit the postsynaptic cell. What they do is they actually allow, chemically, they actually allow negative ions to come. They actually allow chloride, which is a negative ion, to move into the cell, and it actually makes the cell more negative or less excitable. Okay? So what you have going on at your synapses, the whatever 1,000 trillion synapses that are going on right now in your brain, is you have at any given synapse, it's receiving inputs from a certain number of excitatory as well as some inhibitory synapses. And they're fighting it off. They're fighting each other for control of what's going to happen at that particular synapse. One final little important piece is that at most synapses, a single synapse is not sufficient in and of itself to cause the postsynaptic cell to fire its own action potential. It needs help from its friends. So in the neuromuscular junction, that was one thing that was unique about the neuromuscular junction, in that it actually is a really, really powerful synapse, and so one neuron can drive a muscle cell. But in pretty much every other place that you look, a single synapse just isn't strong enough on its own to cause the cell to fire. It requires a collective effort of other compatriot excitatory synapses, confounded by the fact that there's also inhibitory synapses that are opposing that, really literally like a tug of war. And so what that means is that each one of your synapses is acting like a little microcomputer, where it's looking, for lack of a better term, it's looking at all of the inputs coming in and deciding whether the excitatory outweigh the inhibitory and whether there's enough excitatory input for that to fire. So not only do you have 1,000 trillion synapses, each one of those synapses is not simply following a set rule. It's actually working like a little mini computer, and it's basically from microsecond to microsecond, it's responding to its own range of synaptic inputs and then deciding whether or not it's going to fire. Okay? So, what you think about that? And we'll come back.
Thank you for taking your seats, ladies and gentlemen. This is the second half of the second class of Clark Lindgren's class on a course on synapses. Clark Lindgren.
Okay, so now that you have a little more sophisticated understanding of synapses, let's talk about some ways that we can sort of mess with synapses, or what I like to call better living through chemistry. So, knowing that there are both inhibitory and excitatory synapses throughout the brain, if you wanted to get somebody to chill out, to just relax, to not be quite so anxious, what might you do?
Inhibition.
And I'm speaking of drugs. So what would you want that drug baby to do?
Inhibit sensitive.
Inhibit.
Well, you might want to inhibit, but we know there already are inhibitory neurons there, so why not just make the inhibitory synapses better, right? Okay? And that's in fact what Valium does. So Valium is a class of drugs, they're called benzodiazepines, and they actually increase the effect of GABA, which is that inhibitory neurotransmitter. So if you want to, and it's still used today, there's better ones that work faster and all that kind of stuff, but they're all basically the same idea. They help the inhibitory synapses to be stronger, and so they actually therefore calm people down. And I couldn't resist. Valium, of course, became a really popular drug for primarily women, housewives, back in the 60s. And it was really the thing that sort of allowed them to deal with the challenges of I don't want to go into it too much, but the challenges of being a housewife in the 60s, which was really a difficult thing. My mother actually talked later after this all had happened, really got addicted to Valium, like many housewives did. They just started, they were getting it from their doctors, but they just kept prescribing it. And this is now, you've heard this like 3 times now, but this is a great song that came out. This is one of the first hits of the Rolling Stones, and it's about Valium. And it's...
What a drag it is getting low.
You can relate to that.
Kids are different today. I hear every mother say, mother needs something today to calm her down. And though she's not really ill, there's a little yellow pill. She goes running for the shelter of her mother's little helper, and it helps her on her way, gets her through a busy day.
Anyway, you get the idea, right? Great song, one of the best Rolling Stones songs. And so true. So one more example. So Valium, another example, and this came up a few times, but in most synapses there is the mechanism that's used to turn them off is a reuptake mechanism. Most synapses, unlike the neuromuscular junction, don't destroy the neurotransmitter. They just take it back up into the nerve terminal. And one example of that, okay, me do this one more time. This is going to get really old. I might have to. Part of it is I can't. So Prozac and the drugs that are the class of SSRIs, which stands for selective serotonin reuptake inhibitors. So serotonin is a neurotransmitter, and these widely successful drugs that are used for treating depression and obsessive compulsive disorder and things like that, really they work by interfering with the reuptake of serotonin. So the synapses that are using serotonin then sort of end up not turning off properly. They have excess serotonin, and that actually can have therapeutic effects that aren't entirely understood why that works the way it does, but the initial mechanism is true. And I just came across this. Carrie Fisher was a really outspoken, of course she struggled with depression, and was a really outspoken proponent of drugs like Prozac. And her ashes are put in an urn that's shaped like a Prozac pill. And there it is at her funeral. Apparently she had that in her house somewhere, and she really liked her big Prozac pill. So now her ashes are resting in there. Okay. Okay, reset. We're on to bucket class 2, in case you're wondering. Okay, so we're on to why are synapses so special. And well, I sound like the airplane pilot. We're gonna pick up some speed here and we'll get you over one time. But actually, I think I could do this pretty quickly, even though this is a topic that's near and dear to me. I'll try to keep it brief. But so we talked about, you know everything there is about synapses now, and even sort of have an idea of where things can go wrong in terms of, and also can go right in terms of fixing problems. But why are synapses so special? And there's really 2 reasons. The first reason is because they form these networks. And I want, before just glossing over that, I wanted to make sure you all understood why that's significant. So why is it that creating this complex network is such a powerful thing? And I thought of an example, just to give you sort of one example of how a network works. And this is a network that is pretty well understood. It's been worked out since like the 60s and so forth. So how do you make out images in your visual field, right? So you basically, so first important point is your eyes and your visual system isn't anything like a video recorder, even remotely, okay? Strangely enough, your eyes don't look like they were designed by a Silicon Valley tech firm company. They look like they were designed to do one thing only, that is to allow the animal to survive. Amazing. So they're very, very efficient. They don't bring in high-def information like we all assume they are. We assume our eyes are high-def, right? Because how else could there be high-def TVs unless our eyes were high-def? Our eyes are tricking us all the time. They make us think we're seeing in high-def, but in fact they're not. They're actually seeing pretty low resolution, with the exception of a very tiny spot in our visual field that's high resolution. So when I'm scanning over this room, I have the illusion that all of you are in beautiful high definition resolution, but in fact right now it's only one person I can really see in high resolution. If you really think about it, everything else is a little bit blurry, but your brain is just constantly re-updating and giving you the impression. And you can always refresh it, because if you need to get a refresher and see if something happened since you were staring at somebody, you can do that. But anyway, so I could go on and on about that. But the visual system is a really, really complicated system, and what it's doing is it's trying to extract information that's useful. So what is useful for an organism, including humans, to extract from their visual field? Well, sorry for any artists out there, but it's not to appreciate the beauty and the nuances of a sunrise or sunset or the shading of colors. That doesn't seem to be the way the visual system was really designed. It can actually work okay for that, but it's really designed to look for edges, so that people like me don't fall off the edge of the stage, or even the edge of a cliff, or whack my head into something, or I can dodge a flying spear, or whatever it is that I'm trying to do. So our visual system is really trying to extract details, but it starts off by looking for edges. And that actually begins already in the eyes. So your eyes are wired up so that they actually are trying to identify edges, lines. And so if you think about it, they're looking in the visual field, they're looking for lines, and what you want to know is how long is the line, that kind of thing. But also what angle it has. Is it horizontal, is it vertical, or is it any sort of angle in between? And this is how networks work. So, excellent. So the information comes into your eyes and reaches one part of the visual cortex, and your visual processing occurs on the back of your head, called the occipital lobe. So the information, after it travels through the eyes, and already the eyes have already started to recognize these lines, it comes to the what are called simple cells, and they basically just fire depending on whether the stimulus is a horizontal line, or is it some different angle, or is it a vertical line, and so forth. And that's basically what happens at this first layer. Then the cells that are in this first layer, they synapse on cells that are in this next layer, and depending on what those synaptic connections look like, it will cause cells in this complex layer to fire based on more specific features. So it actually starts to put things together. And for example, one of the things you can't really see what's happening here is one of the interesting things that's happening in these complex cells is they no longer care so much where the retina that image struck. They're just actually interested in the fact that it's a horizontal, vertical line. And so if you think about it, that's a really, really complex process, because your visual system, my eyes are right now looking straight ahead. There's all kinds of lines and all that kind of stuff in my visual field, but my head's moving around, right? And so I don't want those all to be shifting around as I'm trying to interpret what's there. And so these complex cells actually take the output from the simple cells, and they actually sort of abstract it a little bit, so that now these cells are only firing if you have these different stimuli. And then you come to the next layer, and the cells in this layer only fire if these complex cells are connected up in such a way that you have a horizontal line and a vertical line. Well, that means you have an angle. You have a right angle. You don't have this, you don't have this. See how that works? So, and then you go further up, and you start putting these together, and eventually you get to the square, which was what the original, or the rectangle, which is what the original input is. And so that's probably one of the best understood ways of how networks work. You have this initial layer that's extracting the most crude information, but then it's extracting more and more details as you go further up the visual system. So that eventually you get to the point where you recognize that it's a square. Does that kind of make it, you kind of see how networks are so powerful? It actually, interestingly, it goes on from here. This was the stuff that was done back like in the 60s, 70s, but we now know that the visual system keeps going, and actually after the information leaves these top layers, I didn't do that, these top layers right there, then the information goes further back into the visual, or further forward in the visual system, and continues to extract more and more details until eventually you reach what neuroscientists have somewhat facetiously called grandmother cells, which are cells, there's like a cluster of cells in your brain that will only fire if you see your grandmother's picture or your grandmother's face. It's a bit of an exaggeration, but not entirely. And so one really interesting example of this came about a few years ago by some neuroscientists who decided to take advantage of the fact that a fair number of, not a lot, but a certain number of patients undergo surgery to treat incurable epilepsy. So epilepsy is this uncontrollable activity that starts usually in one part of the brain and then can potentially spread throughout the brain. And if it spreads too extensively, it can actually be fatal. Most people with epilepsy or some kind of seizure activity can be treated with drugs adequately, but some people are just resistant to that. And so the treatment of choice in that case is to go in and cut out that little piece of the brain where these seizures are beginning. So if you're slated for that surgery, what would happen is you would first get some electrodes implanted in your brain for several weeks so that the surgeon can figure out exactly where the seizure is beginning. So you have to have several seizures so they can see exactly where it's coming, so they know precisely, because the smaller the piece of the brain you can cut out and be effective, the better, obviously. This is not one of those cases where you want to go in and say, well, I'll take out twice as much as brain for the same price. And be like, no. So some neuroscientists realized this was going on, and they said, we have some patients that have electrodes implanted in their brains that are recording their electrical activity. We can actually, if the people will be willing to, we can actually use them in some experiments. And so they did, and one of the fascinating things they discovered is that these grandmother cells actually kind of exist, except the one that's most famous is the Jennifer Aniston cell. So one of the subjects actually had a very small region in his brain, probably not a single cell, but maybe just a few cells. They only fired up when he saw Jennifer Aniston's face and nothing else. So, so these networks keep getting more and more abstract, more and more, in some ways more and more specific, building up more and more details until finally you've got Jennifer Aniston, and there she is. Interestingly, that case, the cell was far enough along the processing pathway that it would also fire if her name was said, or so it actually had integrated with other types of sensory information like visual, like sound, and reading. So actually reading her name or whatever. So, you can think about it, if there's people you know really well, they probably have a region of their brain that fires every time they think of you. Cute thought. No, I'm dead here. I'll skip over that. I want to show you this picture. Okay, one more time and then I'll give up. So remember I mentioned the Human Connectome Project, which is all this millions of dollars that the NIH has been infusing into neuroscience research to figure out that basically what it comes down to is trying to figure out what these networks are, because we understand that that's how the brain works. Individual cells are not allowing you to carry out the amazing things your brain does, it's their ability to work together in these really, really complex networks. And so I wanted to show you, this is a fairly recent paper, came out in Nature, and these experimenters are part of that project, although what they're working on here is just the mouse brain. And after about, I forget exactly how much time, but several years of work, so this actually ended up being at least 3 or 4 years of work in one lab, which is at Harvard, and then some more complex computational work that happened at the Allen Institute in Seattle. They came up with this network, and so what this is showing are the cells and the synapses in a region of the mouse brain that's about 0.5 millimeter by 1/10 of a millimeter by 1/10 of a millimeter. And that took many, many years to accomplish that. And actually, it's from the visual cortex, and so I thought you might find this interesting. So what they're showing here, and so this is actually not just a structural map, but also a functional map. And so the way they did this is they cut put a little window in the mouse brain in the visual cortex so they could actually record what was going on while the mouse was still alive and looking at visual information. And so they were able to compare the function to the structure. And so you see these bars down here? So they're color-coded to show the different cells that respond to different orientations of bars of light. And then they're starting to get an idea then of how those are working. And one of the things that's interesting, you can see that they're all mixed together. So, as far as the way a network works, is it doesn't have to all be nicely segregated into, you do this and you do that and you do this, but they can actually be all mixed together as long as they're hooked up in a way that actually will allow that function to emerge. Okay. So that's, what am I talking about? Why synapses are cool. So they're exciting because that's really where we think the take-home message is going to be. Understanding not individual cells so much, although we have to understand those before we can understand the networks, but it's really seeing how those things are put together in networks. All right. Now, the second reason that synapses are special is because they're plastic. So what does it mean to be plastic? You think of plastic bottles. What does the word plastic mean? So flexible, and specifically like the word came because it meant that you could like take a mold and you could actually create something to reflect the mold, like basically it was flexible, and then it would retain its shape. So it would actually be flexible, adjustable, but then it ultimately would retain its shape after it had actually been modified. And so synapses, the thing that I'm fascinated about is that they just they're not stuck doing the same thing day after day after day like some of us are. They're changing all of the time, milliseconds of time. They're constantly changing. And the way they change can be divided up into two different types of change. So one that I call physiological plasticity, and so that refers to the fact that the synapses can change their function a little bit. And usually what we mean is they become stronger or they become weaker. So remember I talked about the tug of war happening at synapses between excitatory and inhibitory inputs? Well, if suddenly the excitatory inputs get a little bit stronger, in other words, they're either they're releasing more neurotransmitter, they're producing a bigger effect, then they can start to dominate what's happening, and vice versa, if they get weaker and so forth. So they have physiological plasticity, and then there's also structural plasticity. So the synapses don't remain static, but they actually can adjust, they can change their shapes, and those shape changes actually also have effects on their function. Okay. So the strength of the synapse can change. And kind of going back to our earlier picture, they can do that in two different ways. So there are presynaptic mechanisms and then there are postsynaptic mechanisms. And these reflect what we were just talking about. So what do we say that the key players were in terms of the presynaptic events that happen in a synapse? Calcium channels and the synaptic vesicles and their release processes, right? So those can be changed. So not only are those susceptible to disease like Lambert-Eaton syndrome, but they actually physiologically, those calcium channels can be changed. They're proteins, and proteins can be modified in very rapid ways, really clever ways that cells can, so that these channels can now either maybe open up wider so that they let more calcium come in when they get activated. Well, if more calcium comes in, what do you suppose is going to happen? Might trigger more might trigger more of the vesicles to release their contents. So if you basically change these calcium channels so that they open up bigger, they're actually going to cause more neurotransmitter to be released, and so that synapse gets stronger. Okay? Or vice versa, you can actually make these work the other way. And also the release machinery, which we were talking about, the ability for the vesicles to release their contents, that can be changed. It can become easier or it can become harder. And depending on which one of those happens, the synapse will get stronger or it will get weaker. One sort of subcorollary of that is synapses can rapidly change the number of synaptic vesicles. So if you suddenly have fewer vesicles there to be released, all things being equal, you're going to have less neurotransmitter being released. Okay? So those are happening, again, at the 1,000 trillion synapses in your brain. Not only are each of these synapses summing up all of the inputs that are coming in through both excitatory and inhibitory inputs, but those inputs are changing constantly. They're changing in response to things that are happening, and changing the number of synaptic vesicles. The calcium channels are changing. So those are all the things that can happen presynaptically. There also are postsynaptic changes that can happen. So what was on the postsynaptic side? The receptors, so the neurotransmitter receptors, in the case of the neuromuscular junction, it's acetylcholine receptors, but the locks that these neurotransmitter keys are binding to. So those can change. And so how can they change? Well, more, fewer, right? So those can actually, this is the number of receptors, and they can change really rapidly. So synapses can suddenly put a bunch more of those receptors into the membrane, and now, all things being equal, they're going to basically open more of those channels, and they're going to have basically that synapse is going to be strengthened. Okay? So it's all, you know all this stuff, right? Because you know those. It's really not that complicated. It's just the calcium channels, the release machinery, the receptors that bind to the neurotransmitters, and then there's also the inactivation processes, which is the other category. So you can also have changes, although these don't seem to be as common physiologically. So I talked about sarin gas is a way that you can actually pretty traumatically alter a synapse, or SSRIs, the Prozac drugs, those can actually interfere with the inactivation process. That doesn't seem to be something that nature does too often to modify its synapses. It really either seems to be messing with the stuff that are happening to get the transmitter released or to receive the neurotransmitter. Okay. Now, so here's an example. I want you to focus in on, we're going to get a quiz on this next week. Now, the reason for showing this is actually my own work. This is the stuff I do, and so I'm interested in those physiological changes that happen to synapses, and I'm just wanting to show you how complicated it can be. Okay. So there's a few things on here you should recognize. So this is the presynaptic terminal, which I drew in this paper. These are the synaptic vesicles that have the neurotransmitter acetylcholine in them. So everything's okay so far. These are the receptors that acetylcholine binds to and opens up to activate the muscle cell. And all this other stuff is just actually one bit of the regulatory proteins and processes that occur to change it. So you have to, so you remember that. So we'll have to really work on our eidetic memory. So you can all remember that, right? So you have all these things that are happening. The basic process I showed you is pretty straightforward, but then Synapses invest an incredible amount of effort into creating mechanisms for altering what's going on. So that is what really makes Synapses, in my view, really kind of special and exciting. Okay. So that's to kind of give you just a very general idea of what we mean by physiological changes. What about structural changes? Well, so now if we're looking at, so this diagram here is showing a cell that's been labeled with this fluorescent dye. This is the cell body. So remember, all neurons have the cell body. This is where the nucleus would be and so forth. And these are the processes that extend out. And these specifically are called dendrites. And this is where this particular cell will get synapsed onto by another cell. So all of its inputs are coming in through its dendrites. And if you look really closely at these dendrites, remember that? They had these little spines sticking out. You saw those? Those are actually called dendritic spines because they're these little prickly things that stick out of the dendrites. And those spines are where the synapses happen. So those 1,000 or so synapses that are coming, they don't synapse onto the dendrite itself, they synapse onto those little spines. Okay. Well, why is that significant? Well, because it turns out that those spines can have their shape changed really rapidly. So they can actually, so this, I'm just going to show you this. This is showing a synapse onto a spine. So here's the dendrite down here, and you have the spine sticking up here. This is where the glutamate receptors are. And this is the presynaptic cell that's synapsing, one of the 1,000 or so that are synapsing onto this postsynaptic cell. And it's releasing its glutamate, binds to these receptors, and then the information then goes to the dendrite, then travels to the cell body, and then eventually to the axon to cause the cell to fire. So by changing, so you can kind of imagine, how that, you're really going to have to imagine how that works, but how the dendrite spine might change, right? So it might get bigger, or it might get smaller, it might shrink, it might enlarge, and in fact, they're doing that all the time. So these little spines are changing their shape. Mostly they're getting bigger, they're getting smaller, and when they get bigger, they have a bigger influence on the postsynaptic cell. When they get smaller, they have a much less influence. So here's this is a good place to kind of end. So here's a really exciting, I want to give you a couple of examples. I'll give you another one next week. But this is an example of the really exciting work that's being done now in neuroscience using optical tools. And so I'll just show you the experiment now so you can kind of get an idea how that works. And then I'll tell you a little bit more about it next week because that's going to be part of some other things I'll tell you about. So what this is showing, this is a paper that's showing, so here's a mouse, and it has a little window cut in its skull so that the investigators can look at its neurons while it's alive and while it's functioning. So this is just a little cutout that's showing what some of those cells look like. And so this is the cell body, and this is the dendrites of that cell, and these are the little synaptic spines. And then the blue are showing the synaptic inputs onto this cell. All right. So the question is, what happens to those spines when the animal learns something? What happens when it becomes better at doing the task? And so they injected what's called a memory probe. So they used a genetic trick to basically become expressed in those cells that, well, let me come back to that. Let me go down here to this thing first. So the mice were taught how to run a rotorod, which is, the best way of thinking of that is it's kind of like, the log roll. you know. And so there's a skill involved. So the first time you get on a log and you try to basically stay up as long as you possibly can, and you fall into the water or whatever, and everybody laughs. But after a while, if you did that, you start getting better at that. So it's an example of sort of learning sort of motor memory. And so what they did is they actually visually, literally looked through this window in the mouse brain at the dendrites. And this is what they looked like before. This is a cartoon of their data, but this is basically what it looked like before they learned. Okay. After learning, notice what happened. They got more spines, and some of the ones that were already there got bigger. So like this is a new spine, looks like this one, I can't tell. But basically what happened is the spines got bigger, and there became more of them after the animal had learned the task. What they did, though, the really clever part is they put this probe in so that in those cells that had been, and in those parts of the cells that had been activated, they started expressing this protein which they could activate with light. And basically, usually the way these experiments are done is you will introduce light. I think in this case, they just actually introduced the light through the window. But in other experiments where similar techniques are being done, is they take a little cannula and snake it down into the brain a little bit further down, and then hook it up to a laser so they can actually shine really bright light at a very specific spot. But what this light does is it causes these synapses that had been activated to degrade. Does that make sense? So this is after learning, but then they took half of their animals, let them be. The other half, they shined light on them so that the synapses, that the spines shrunk, either went away or got smaller. So the question now is, how's the animal doing on its rotorod skills? Okay, and that's shown right here. So the way it's a really simple, the same way you would grade somebody if you were watching them do a log roll, you'd say, how long do you stay up before you fall in the water? And so, same thing, so this is seconds, how much time they stayed on the log before they fell off. And when you get better at it, you can stay on longer. And so what happened is, before learning, they could stay on about 200 seconds. After a practice period, they could double their time. They could stay on about 400 seconds. If you did nothing to them, if you didn't introduce this probe, they could continue to do that. They continue to be able to have that skill for actually quite a long time. The ones that had been zapped with this light, they lost the ability to learn that. So they went back to being about as lousy as they were before they started. Right? So that's kind of the brave new world of neuroscience, right? Think about what these investigators have just done. They've first observed a physical change in the brain of these animals after they learned something. Okay? But then even more impressively, by doing something completely foreign or alien to the animal, by reversing that change that had occurred, the animal lost that ability to learn. Okay. So very, very, very specific, very, very precise way of inactivating that learning process. And by simply manipulating the physical elements of the brain. And so going back to the first lecture where we were talking about the mind-brain conundrum, the animal, by simply manipulating the physical parameters of its brain, could be made to, we could see those changes, but then we could also, like I didn't have anything to do with this, but those changes could also be reversed. And so that's kind of really where neuroscience is right now. And we have a little more, 5 minutes. I'm going to introduce the next topic because we're going to have a little bit less of a class time. So I've explained what a synapse is, why they're special, again, because they can form these networks which allow them to collectively do these amazing things. They're plastic, they can adjust both physiologically and structurally. So then how do the synapses make us? And you're kind of probably getting a little bit of an idea of that because of the example I just showed you. But there's a lot of things we could look at. At the beginning of class, we talked about what is the self, and there were some various ideas: personality, your emotional, let's say, sort of your emotional intelligence or your emotional environment. But I think that, and I had to pick one because we don't have time to sort of go through a lot of the different experiments. But the one to me that is most significant. I forgot what was up. Is memory. I remembered. And what else? You know, if we lose our memory, we've really kind of lost ourselves. I mean, we always have sort of a new beginning, a new sort of identity. But I really think, as this quote from Luis Buñuel says, you have to begin to lose your memory, if only in bits and pieces, to realize that memory is what makes our lives. Life without memory is not life at all. Our memory is our coherence, our reason, our feeling, even our action. Without it, we are nothing. And I tend to agree with that. It might be a little bit extreme, but I sort of feel like most of us feel that our memory is really sort of a big deal. So what we're going to focus on in this next class is specifically how that happens. And maybe that's, well, there we go. All right, so we'll start on that. That'll be a good place to start next week. So we'll start looking at what neuroscientists know about memory and how that comes about.
Thank you very much, Clark. We learned a lot this morning, and I think the secret now is to stay away from the blue light. Isn't it incredible that if anything works at all, when you see how complex things are? Really appreciate your information on this part.
Considering an iPad can't even work, how can our brains do that?
We will see you all next week at 9:30 class. So come a little after 9 and we'll get you all checked in. And then class will end at 10:45. See you next week. Thank you so much for coming this morning.